I'm delighted to be joined by Professor Robin Shattock, who is the head of mucosal infection and immunity within the Department of Medicine at Imperial College London. Robin's research focuses on the mechanisms of mucosal infection and the development of novel preventative strategies, including pandemic vaccine responsiveness and the development of vaccines against HIV, Ebola, Marburg, Lassa fever, and rabies, all appropriate to a developing world setting. Last year, he also co-founded and is now CSO of the Imperial spin-out Vax Equity, which, in a nice segue from the earlier fireside chat I had with Semeni Pangalos, earlier this year, the company signed a deal with AstraZeneca, potentially worth up to $195 million, with a focus on the potential of self-amplifying RNA as a therapeutic approach. So, Robin is well placed to discuss the opportunities around that RNA gives us as a therapeutic approach. So, Robin, thanks very much for joining me.
A pleasure.
So, we've seen the impact of mRNA technology for developing vaccines against the pandemic. So I was just wondering, from your perspective, to what extent, was this something that actually you anticipated or expected could happen?
Well, I think we were anticipating that there was a pandemic, and I presented at the World Economic Forum in 2019, suggesting that RNA would be well placed to respond because it can be manufactured so quickly. We didn't know that COVID was just around the corner. And of course, it was a technology that had been developed for quite some time, and it just happened to come of age at the right time point.
Yeah. So, sort of looking at what happened, what are the most important lessons we've actually learned from the sort of COVID-19 mRNA experience?
I think it was very interesting in that most people wouldn't have expected that mRNA vaccines would be the ones that were developed first, who weren't in the field. I think one of the things that immediately became obvious is this technology had been being developed in the oncology space. And so nobody had actually thought about the complications of making a vaccine that needs to be globally available. So that the production capacity to make hundreds or even billions of doses was not there. And the stability issue had not been solved because it hadn't been a major problem for the oncology space.
Right. So, what is it about RNA? You sort of mentioned at the World Economic Forum that you sort of suggested that it would be a potentially good approach. So what is it about mRNA and those RNA therapeutics that actually makes them an interesting target?
I think for two things. In the pandemic space, essentially you're working with genetic code. So rather than needing a complicated manufacturing footprint where you're having to make recombinant proteins or viral vectors that take that much longer and may require liters, thousands of liters to produce millions of doses. This can be scaled down in a much smaller volume in a fully synthetic way. So it kind of changes the way you manufacture. And of course, it has the flexibility that you can encode anything you like in your vaccine construct.
You mentioned that RNA or mRNA was first being pursued in other indications in the oncology space. I think also, in fact, people like BioNTech were also looking at in the sort of infectious disease space. I think they originally had a deal with Pfizer, pursuing flu. So I just wonder, what are sort of the prospects beyond, COVID-19 for the mRNA as a modality?
Well, clearly everybody's going to be going back to focusing on oncology, personalized medicine. But I think it's opened up infectious diseases as well. But the challenges are to reduce the cost and make the stability better. And I think that's important going forward. And then people will be looking at as many indications that it can be applied to. Now, it's not a panacea. It will work for some things. It won't be fit, for example, for working against bacterial targets that need glycoconjugates, as an example. So you won't see a meningococcal vaccine necessarily using RNA technology.
Right. So, clearly, the challenges of almost like that first generation of mRNA vaccines, so stability. We saw that, the issues around the cold chain, the fact it had to be kept at minus 80 or thereabouts. What will the next generation of mRNA or mRNA-like vaccines and therapeutics look like?
Well, we think the next generation needs to be bringing the dose down. We think self-amplifying RNA is one of the ways to do that, so that one can realize combinations. We need heat-stable, easy-to-use formulations, thinking about different routes of delivery. So, moving away from always using injectable vaccines, also thinking about mucosal or transdermal approaches of delivering the RNA cargo. So it has enormous flexibility for the future.
Right. And self-amplifying RNA, can you explain a little bit more what that's about?
Yeah, so it's like RNA, the kind of first generation RNA, except it has built into it this capacity that it can copy itself when it gets into target cells. That copying doesn't go on indefinitely, but it goes on for a period of hours to days. And it means that you can use potentially much lower doses. And that's important for a number of reasons. Firstly, it means you can just produce more vaccine. It also means that when you're thinking about pediatric populations, it means you have a much higher safety margin, which is probably going to be important. And also for combinations, our target dose is a 1 microgram dose. So if you were going for a quadrivalent vaccine, it would be 4 micrograms. Moderna currently are at 100 micrograms. You can imagine 400 micrograms is going to be a big challenge. And then perhaps you think about different routes of delivery. If you were thinking about a microneedle approach, you might lose, a significant amount of your cargo just in that delivery because it's more inefficient. Well, if you needed 10 times the product, it would be easier to do for self-amplifying RNA. And the final characteristic, which is really interesting, is the kinetics of expression. So RNA is expressed very rapidly. It peaks at about 24 hours and is gone by about 72 hours. Self-amplifying RNA peaks at about 5 days and can go out for weeks. And so for biotherapeutics, it may be more interesting in terms of if you could move to a monthly therapy rather than something that might need to be done every few days.
Right. And the company that you co-founded, Vax Equity, it's built around the use of self-amplifying RNA. I'm just wondering, what was it that you were able to, for example, show AstraZeneca that actually convinced it to sign such a potentially lucrative deal with Vax Equity?
Well, I think there are a number of aspects. First are the kind of differences that I've already discussed in terms of the potential advantages of the approach. And also they look very carefully at our aggressive development plan. And if you like, our first clinical trial was, taking a prototype that was built out of an academic setting, grabbing things that you could pull together very quickly with very little optimization because it was COVID-19. And so we've got an enormous scope of improving, designing the technology to make it really fit for purpose. To some extent, self-amplifying RNA is about where mRNA was 5 years ago, before it had many clinical studies.
Right. And when the deal was announced, I think it was 26 undisclosed targets. Now, I know you're not necessarily going to then start telling us what those targets are, but what sort of therapeutic indications? What sort of diseases? Is it just infectious diseases, or is it, as we heard from many earlier on, they're into respiratory, they're into oncology, they're into immunology? So could you give us some sort of idea of the scope of what you might be targeting?
Well, obviously, I can't tell you exactly what we're going to be targeting, and in terms of the partnership with AZ, that still needs to be fully defined. But essentially, it has broad applicability across all those areas of interest that AZ has, and so I think we'll see that it's likely to be used for as many different types of indications, and some of those may work, more easily, maybe lower hanging fruit than others that may take a longer time to really optimize.
Yeah. In my introduction, I mentioned the fact that, your research interests are, very much, around, HIV, Ebola, Marburg, Lassa fever, etc. And, particularly, diseases that are almost like have a massive impact in the developing world. I just wonder what optimistic sort of noises can we make about, for example, the RNA approach to tackling those diseases, which are clearly a massive, still unmet medical need.
Well, I think that they will change the way we work, and I think they will accelerate things. But we also need to recognize that to some extent, we were lucky with COVID-19. I know that sounds strange, but it was a relatively easy to neutralize virus. And something like HIV, I've been working in that field for 30 years, is a much more complex target to tackle. But the RNA technology also allows us to get things into human studies much more easily at lower cost. And so it allows us to do more of that discovery process through human trials rather than relying on animal models, which have not always given us the right answers.
Right. And so what are the sort of the time frame, do you anticipate for, these new platforms to provide real benefit to patients?
I think we'll see them providing benefit in the next 5 years for sure, and beyond. I think it will, become more and more dominant.
And can you, without sort of like giving away anything that with the AZ, what Vax Equity itself, what are you actually focusing on? I mean, and where would you want it to have an impact on patients?
So Vax Equity is focusing very much on the infectious disease space. So they're focusing on optimizing the platform and going for targets that are really relevant, and initially those that perhaps are not too challenging for the technology, but then branching out to harder to reach targets. But again, I'm not in a position to reveal what those targets will be.
Right, but and so the time frame, when do you sort of anticipate getting sort of like at least first into man?
Well, we've already first into man through the academic route, and we have a second generation vaccine in trials. So I think we'll be back into the clinic fairly soon, but we may go through a range of iterations through clinical evaluation to fully optimize the platform.
Right. And the self-amplifying RNA, you sort of said that it makes copies of itself, but it isn't something that goes on ad infinitum. That there is a certain, what is that a sort of a switch that you have engineered into the molecule, or is it just a natural consequence of the ecosystem within the body?
Well, it's a natural consequence of the platform. So it's based actually on a virus. So most people use an alpha virus, and it uses the non-structural proteins that can do this replication process, and they naturally self-limit themselves because built on a virus wants to replicate and get out of cells. And not stay there for indefinitely. So over time, it switches from copying the whole RNA to copying the component that encodes your gene of interest, whether that's the spike glycoprotein of coronavirus or whatever you've encoded. And then it just runs out of steam after a period of time. So it naturally self-limits.
Right. And I mean, it sounds like this is a platform technology. So is your business model to license this out to in indication specific ways to other partners?
I think at the moment we've got our hands full. So we're not looking to license it out. We're looking to develop it and exploit it fully.
Right. And again, sort of because of your focus very much in terms of almost like vaccine equity, to actually get it into the developing world. What are the sort of the prospects for these self-amplifying RNAs? I mean, in terms of like stability, do they have to have that sort of cold chain the way that the mRNA vaccines have?
I mean, that needs to be changed, and we're very much focused on changing that, but I think all the RNA companies are focused on changing that. We already believe we can realize a vaccine that can be stored at refrigerated temperature, and we will be looking for ways to store it at room temperature. We're not there yet, but I think that's really a solvable problem. It's not a major challenge. It just hasn't been, focused on because people have just been moving at an exceptional speed in the COVID space.
Yeah. And I mean, have you sort of looked at the technology in the COVID setting?
We have looked. I mean, have we looked at self-amplifying RNA in the COVID setting? Yes, absolutely. So we've taken that into a first clinical trial. And we're taking a second generation approach into a second clinical trial and looking at how well it performs. Now, it's a crowded field right now in that there are many vaccines that have worked and already been approved. So I suspect commercially it's not going to be sensible to be going for a first generation COVID vaccine. Whether there will be a commercial sense to go after booster regimes or combination boosters with other vaccine indications is something that we're considering. Right.
So whether that would be, I mean, could you create a sort of a COVID vaccine that actually covers various variants, sort of the Delta variant, the Lambda variant, etc., or are you sort of thinking about, combining it with, say, for example, flu vaccine, because we know that, for example, there's flu vaccines are given to senior citizens,
right,
to protect against flu season. So, is that something that's possible?
And it's certainly possible. I think many companies are thinking about a flu COVID combination product. It's more challenging the higher the dose you need to use. So in that respect, self-amplifying RNA may have some advantage. The question about variants, everybody is also interested in variants. I don't know that you necessarily need a combination of variants. We're still looking at how that kind of variant geographical distribution goes and whether it actually settles on something that can be targeted individually.
Right. So Robin, it's been fascinating talking to you. Really interesting to hear about this, self-amplifying RNA, and I look forward to sort of reading more about the successes, etc. So, thank you very much for joining me today.
My pleasure. Thank you.